Hello students, welcome back. Today we will be discussing the classification of organisms into three main domains, that is bacteria, archaea and eukaryotes or eukarya. So the word given in the learning outcomes of the syllabus is describe. So we will be describing the three domains. It means we will be talking about the main features or the main characteristic features of each domain. So let us start. We have three main domains mainly, and when we say domain, it means the highest taxonomic rank. It means the living organisms have been divided into three biggest groups, which we are calling them domains. So it's the highest taxonomic rank in the hierarchical classification. And these three domains are bacteria, archaea, and eukarya. Bacteria and archaea, they are prokaryotic organisms. By prokaryotic organisms, we mean they do not have nucleus in their cells. It means they do have DNA or the genetic material or genome, but they are actually lacking a nuclear boundary or nuclear envelope, which otherwise we see in eukarya or eukaryotic organisms. So bacteria and archaea, they are similar in the respect of lacking nucleus in their cells. So in this way, they are actually similar, but they are unsimilar in many other ways. They will not be similar in many other ways. So let us see what is that. Eukarya, these are the living organisms which have nucleus in their cells, and they're the main kingdoms or the subgroups, you can say, of eukarya are protists, fungi, plants, and animals. Okay, we will be talking in detail about all these three domains, but let me tell you one very important and a special thing about bacteria and archaea. In fact, bacteria and archaea, as I told you, they are similar, that they do not have nucleus, and they are similar in certain other features as well. But what is the big difference between bacteria and archaea is that bacteria, they are living under normal environmental conditions, but archaea, they they are not living in the normal temperatures. Rather, they can withstand the harsh temperatures or very high temperatures. So that's why sometimes you will be finding that you will be given this word as well, extremophiles. Extremophiles. So the organisms which can live or which can withstand the the very high temperature or harsh temperatures, we can say. So let us see. Now we have to move to talk about these three main domains one by one. I might come back to show you these pictures after a while. Okay, so first of all, characteristic features of bacteria, and bacteria domain we also call eubacteria, or we also call them true bacteria. These are the bacteria which is the real bacteria, because the characteristic features which we are going to talk about, they actually they have those all characteristic features. So let us talk about. Number one, they do not have nucleus in the cell, as I told you. Number two, DNA is circular. DNA is not linear, it is circular. It's like a loop. It's like a loop. For example, if I can draw over here, it's like a loop, like it's end to end join, like a loop. And it does not have histone proteins. Histone proteins are very important proteins. They are the proteins which actually give the packaging condition to the DNA molecule. So these, because DNA of bacteria is thought to be not very long, and it is circular. That's why histone proteins are as such not needed to make it so much packed. Plus, there is no nucleus, so the DNA is just lying in the cytoplasm in without a nuclear boundary. So histone protein is absent. Small circular molecule of DNA, plasmids, are also present in bacteria. Not all bacteria, but most of the time they are present. Now, if I can simply tell you a simple bacterial cell, so you can have main DNA, you can have main DNA, and then you can have small circular DNA, which can be small and many. So we call them plasmid. I'm sorry, I think I should erase this part. Okay, so plasmids are also present in bacteria. As I told you, not all the bacteria, but many of the bacteria, or rather, I should say most of the bacteria, they contain plasmids. So there are two types of DNA or the genetic material in bacteria. One, we can say the main DNA, which is in the form of a loop, or we call it circular bacteria because end to end is joined, and it does not have histone proteins attached with it because no much packaging is needed in case of bacterial DNA. The other extra bacteria, we call them plasmids. They are also present, and many of the bacteria they contain it. And this is plasmid which we actually manipulate in genetic engineering. Okay, now next point is no membrane bound organelles are present. It means mitochondria, endoplasmic reticulum, Golgi body, chloroplast are absent in bacteria. Simply, no membrane bound organelles are present. Ribosome, as you know, ribosome is a very important organelle. It is responsible for making proteins. So what is the size of ribosome? It is smaller than in the eukaryotic cells. Okay, it is smaller as compared to the plant and animal cell, and it is 70S. And you know, S is a Swedberg unit. Cell walls are always present in bacteria, and they contain peptidoglycans. There is a carbohydrate which is not cellulose, and you know cellulose is found in plant cells wall, but in bacteria cellulose is absent and peptidoglycan carbohydrate is present because their cell wall is made up of peptidoglycan. Okay, cell divide by binary fission. Very important, very important. There is no mitosis. As you know, mitosis has many stages like prophase, metaphase, anaphase, telophase. But in case of bacteria, you will not be finding mitosis. One bacterium can be simply cleaved into two by the method of binary fission. Usually exist in single cell. So usually we'll be finding bacteria in single cell or sometimes in a small group of cells also present. So this was all about bacteria, characteristic features, eubacteria or true bacteria. Okay, now let us talk about archaeans. Characteristic features of archaeans. So we have to describe archaeans or archaea. You can say archaea also, or you can say archaeans as well. Okay, so no nucleus. No nucleus in the cell. This is definitely matching with the eubacteria or bacteria domain. DNA exists as a circular chromosome and does have histone proteins associated with it. So it's circular chromosome, DNA as a circular chromosome, and it has histone protein associated with it. This is the characteristic property which is not matching with bacteria. So histone is present in archaeans, which is actually needed for the packaging of the DNA. It means the circular DNA of archaeans does need histone proteins for packaging. Next, smaller circular molecule of DNA plasmids are often present. This is again matching with bacteria. As I told you, plasmids are thought to be extra DNA molecules, which most of the time present in bacteria, and even it is present in archaeans as well. But before going further, let me write one word for archaeans, which we call what we say extremophiles. So we also call them extremophiles, as I told you, because they can withstand the harsh environmental conditions, especially the high temperature. So there are many those bacteria which are found in at high temperatures. We call them extremophiles, and we put them in archaeans. Cells with no membrane-bound organelles. So here also you will not be finding the membrane-bound organelles like mitochondria, endoplasmic reticulum, Golgi body, chloroplast, etc. as like bacteria. Ribosomes are 70S, and the size of the ribosome is again matching with bacteria domain. They are smaller in size, but very interesting thing is the small subunit of ribosome is more similar to eukaryotic ribosomes. It's not matching with the bacterial ribosome. So if you see this point, ribosome point, which is actually 70S, is the characteristic feature which is actually half of it is matching with bacteria domain, and the other half is matching with eukarya domain. So very interesting. The base sequence of ribosomal RNA and the primary structure of ribosomal proteins are more like those in the eukaryotes rather than bacteria. So, as I told you, ribosomes, although they are smaller in size, and what is the size? It is 70S, which is like bacteria domain, but the other half, which is saying that the base sequence of the ribosomal RNA and the smaller subunit of the ribosomal RNA, and the primary structure of ribosomal proteins, they are more matching, or they are more like those in the eukaryotes rather than bacteria. So very interesting thing.